Now that we have discussed amphibian fertilization, cleavage, and gastrulation, we turn to discussing the mechanisms that are important for the specification of the amphibian axis. This fate map of the amphibian oocyte shows that the animal hemisphere will give rise to the ectoderm, specifically the epidermis and the nervous system, while the cells of the vegetal hemisphere are going to give rise to the endoderm. The region at the equator, which is sandwiched between the presumptive ectodermal and endodermal tissues, will give rise to mesodermal tissues. The organization of the different germ layers involves inductive signals that are coming from the vegetal cells. Now, these vegetal cells not only differentiate into endoderm, but they also induce the cells that are immediately above them to become mesoderm. And the underlying mechanism for that specification involves the mRNAs that have become localized to the vegetal hemisphere and gradually specify the different germ layers. Now, these maternal mRNAs include the mRNAs for the paracrine factor VEGH1 and the transcription factor VEGT. These are deposited into the oocyte before fertilization, but are translated after fertilization and apportioned to the vegetal cells during cleavage. Now, the maternal VEGT is important for activating a second set of transcription factors that is not shown here, that specifies the cells in the vegetal region, the most vegetal region, to become endoderm. Another gene that's activated by VEGT is the paracrine factor nodal. And nodal is going to be secreted from these endodermal cells and signal the cell layers above it to become mesoderm. Now, some of the genes activated by nodal also lead to the zygotic activation or zygotic expression of VEGT in the presumptive mesoderm. So VEGT is not only important for specifying the endoderm, but also for specifying the mesoderm. So if we were to get rid of VEGT, we would have no endoderm and no mesoderm. So basically, what we would end up with would be an embryo that it's just a ball of ectoderm. Another maternally derived mRNA is VEGH1, which is localized to the vegetal cells, but specifically to the future dorsal region. And this paracrine factor is required to activate genes in the dorsal mesoderm. So it's going to activate genes in this dorsal mesoderm region. Now, since nodal and VEGH1 are needed for the mesodermal phase, so to induce the mesodermal phase, if we were to knock down these genes or if we prevent the signaling events, we basically lose the mesoderm. Now, the gradual activation of these different sets of genes is what leads to the specification of the three germ layers by the late blastula stage, with the cells of the animal hemisphere, which are separated from the vegetal hemisphere by the blastocele and are not exposed to those signals, becoming specified as ectoderm. The localization of maternal factors is also important for the specification of the dorsal ventral and anterior posterior axis. But in this case, the axes of the body are specified as a consequence of fertilization, and they become fully apparent at gastrulation. Now, recall that the dorsal ventral axis is specified by the site of sperm entry, with the site where the sperm enters becoming the belly of the organism. The specification of the anterior posterior axis is linked to that of the dorsal ventral axis, and it relates to the localization of the transcription factor beta-catenin to the site that will become the future dorsal region of the organism. The localization of beta-catenin to this presumptive dorsal region is needed for the involution of the mesoderm during gastrulation, and that process serves to establish the anterior posterior axis. For example, in the central nervous system, the specification along the anterior posterior axis involves the induction of the ectoderm to form different nervous system structures depending on which mesodermal cells it receives signals from. So the first mesodermal cells that involute into the embryo will induce the ectoderm above them to produce anterior neural structures like the forebrain, which is the most anterior portion of the brain. On the other hand, the mesoderm that involutes later will signal the ectoderm to form more posterior neural structures like the hindbrain and the spinal cord. Now, the induction of the nervous system by its interaction with the underlying mesoderm has been termed a primary embryonic induction. But as we'll see later, the specification of the neural fate is not that simple because in amphibians, the neural fate rather than the epidermal fate is considered the default state for the ectoderm. I mentioned that the specification of the dorsal ventral axis is the result of the events that happen at fertilization, specifically the cortical rotation. But there are experiments that have shown that the specification of cells in amphibians is largely conditional. One of these experiments is the one shown here, which was carried out by Hans Spemann. Now, in this experiment, he took an early nude blastula after fertilization, and he separated the zygote along the first cleavage plane. So along that first cleavage plane, and if you recall, that is the plane that's going to separate that gray crescent amongst the two first blastomeres. He then constricted the egg so that nuclear divisions would only happen on one side of the embryo. And after a period of time, sometimes as late as the 16-cell stage, one of the nuclei would manage to escape the constriction and move into the region that did not previously house any nuclei. At that point, cleavage would begin at that site, and when the two sites were fully separated by pulling tightly on that hair, two genetically identical larvae would develop, with one being more developed than the other as depicted in this image. Now, from these results, Spemann speculated that the nuclei of the early amphibian embryo were equivalent and thus were capable of giving rise to normal larvae.